Assalamu alaikum. Hello everyone. In this tutorial, we will talk about how to perform homology modeling using modeler. And so for that, first we will go through the background in one or two slides to make it easier for you to understand the homology modeling, and then we will jump to the modeler. So, what actually is homology modeling? Homology modeling refers to constructing an atomic resolution model of the target protein from its amino acid sequence and an experimental three-dimensional structure of a related homologous protein. That is the template. So here, actually, the target protein is the protein which we want to use for constructing a structure, and we select the templates of the that are the homologous proteins, or they have similar sequence with high sequence identity. We select the structure of these homologous proteins and model or build the structure of the target protein. So what are actually the major steps in homology modeling? Number one is actually we need to find the template, so that whether there are any specific templates or good templates which are available in the protein database for our query sequence. And for that, we align our sequence with the database of the potential templates, and then we find the best templates with good alignment, and then we find the structures of those templates or the sequences which are aligned with our sequence, and then we build the model using the homology modeling. And we will go through the steps that how we build the model later on, and then after building the model, we actually evaluate the model, that whether the model which is predicted is good or for further use. So for that, we will use modeler for homology modeling, and we will go through the different details. For example, what we will discuss here mainly, that how to install the modeler. It's very simple, but after installing the modeler or downloading the modeler, we need to arrange the files that to run that modeler for homology modeling. And after running the modeler, we will also discuss that how to predict the structure using the homology model, using the modeler. So these are the few details which we'll go through one by one, and by the end of these tutorial and discussion, we will be able and will be equipped with dealing with modeler ourselves. Let's go and start from the beginning. So let's start. First thing what we need to do is to go on the website and write the modeler, and this modeler will help you to go to the website where the modeler is softwares are located. And then from the download and installation, you go and depending on your operating system, you select the software files, and it will start downloading once it is done. So I've just paused it because I've already downloaded it, and two things you need to do. One is to fill up the form for registration and select the right options, like I also want to use the web service and Microsoft Windows I'm using, and put your institutional email address and then click agreed and accept it. And then it will send you an email on your required on your email address, and you can use the license code and start running the modeler. So now after downloading that, you just go and find the right folder where your modeler is selected, or if you are using this Windows, you can just search the modeler and it will open this command prompt file. You just run it as administrator, and here you can see that now this is the folder. Here you can see that it's written that it is recommended to download the Python. So first let's go and download the Python, and for that you just write Python in the search bar, and that will take you to the website of the download Python, python.org/downloads, and from here you can download the latest version. Again, it's already installed in my computer, but if you want to look, you just download it and click it, and it will take you to that exe file, and here you can see that I've already installed it. That's why when I will try to run it, it will say that if you want to modify it, repair or uninstall, but I'll just cancel it. But if you have not installed Python, it will go to the installation, right? So now these two things are ready, and two things what we needed, one was the modeler and was the Python, and then this is the uh, command prompt file, and then now let's go and find the query sequence or from the database, right? So for this tutorial I have selected a protein with the name Titin, Titin, right? So I am selecting an NCBI protein database and I'm writing Titin, and now when I'll search it, it will take us to the results, and from here I select this amino acid sequence of Titin protein, yeah, and yes, we can download the FASTA format of this protein by clicking the file, create file, and now it is downloaded, right? So I'm downloading this for the later use, I'll let you know, and as well as we can insert that sequence in the BLAST GUI, where we will run the BLAST. Here you can just go on the text file of that FASTA sequence which we recently downloaded, select the whole sequence from start till the end. You can do it slowly like me, or you can just click Control All and you can do it. So after selecting the sequence, just copy the sequence, right, and then go to the website here, and you can just paste that sequence here in the BLAST portal, where there's written that enter the query sequence. After clicking the sequence, just write the name, job title, that is the Titin protein. So, and then important thing is we want to select the PDB database, so that because we want to find the template structures, so we will only look for those proteins which have the PDB structure, so that we can use it in the future, which are similar to our protein sequence. So after clicking that, just run the BLAST. It will take a few seconds, and let's wait. Here you can see that now it will be yeah, now it's almost done. So from here you can see that this is the BLAST result. I have also discussed it in detail in my tutorials, but for better understanding of this tutorial, you can see that this is just an example. So here we have searched for the database, and here you can see that we have found number of protein sequences and their structures which have 100% sequence similarity with our query sequence, and 100% sequence identity, right, with a very high significant E value. So not high, we can say E value is very low. The lower the E value, the higher the significance is. So now what we will do is we will go to the RCSB PDB database, that is the protein data bank database, and we will download the structures of these potential templates. And in this example I will download 4 templates. You can select more than 4 or less than 4 depending on your a question of interest. But here I'm just, for example, I'm using 4 templates. So first I'll go here and click the download PDB file of first template, and I'll repeat the process for the other 3 as well. Right, so first one is this, and the second one is 5JDD, and yes, and we can just click the download again. Now yes, this one is downloaded. Now the third one, 3LCY, that's right here, 3LCY. And then the last one, that is 2ILL underscore A 2ILL, right? Now this one. So now we have 4 different structures, and we will use these 4 structures as a template, and we will perform homology modeling, and we will find that the best possible template for the sequence we are using, right? That is the best possible structure. So now we have 4 files. Number 1, 4 files of the template, and yes, and one is the query sequence. Now one more thing which we need to do is you will go to the documentation, and you can see here that in one example that how we have to make the format of our input files. Now we have all the files ready. Now we will make the input files which are specific for use to begin modeler for homology modeling, right? So for that, you go on the documentation, click tutorial, and here you can see that basic modeling example. You just click that, and from here you just download one example to see how the input files are arranged. So number 1 is the sequence file, right? So for sequence file, it should be like this, that FASTA format, like greater than sign, and then anything you want to write, and then the whole sequence, and at the end of the sequence there should be one steric, so that modeler will understand that the sequence is finished after this steric, right? So I'm just rewriting the name of the text file, that is the query sequence, and I'll copy that again right from here. Let's do it. I'm going through each step very slowly and in detail, so that you do not find any confusion when you will reproduce the results for your own interest, right? So here in the query sequence, you just paste the sequence, and now we need to format the sequence. So for that, there should be a greater than sign like this, and any description, and then those zeros. So after writing the query sequence, in the here, just go at the end and write the steric, so that this will be in the format, and just save it, and just minimize the option here. And now one more thing you need to do is to change the extension. So homology modeler actually use the star.ali extension file of the query sequence. And now what you need to do is to place these files into the into the folder where they are installed. For example, here you go on the C. In my computer it's in C users 3521 modeler 925 in the in and then go into the folder of bin. Here you make one folder where you will put all your files which we will use for the homology modeling. So now I have written modeler 1, and now I will go back on the desktop where all the files are placed. Here I will select the query sequence file and the uh, 4 structure files and the pdb underscore 95 PR file which I downloaded while I was downloading the homology modeler. So I'll just copy those files. It's not showing copy. Why it's not showing copy? Just now, okay. So I'll just copy those, this one and that one separately. First, just go here on the not this one, bin folder, and then modeler one, double click it and just paste the query sequence.ali file, and also the rest of the files, right? 4 template files and the pdb underscore 95.pir file, right? So just click these, this one, and the 5jdt, these 4. So we have 4 files. The total number of files is 6 input files which we will be using in the modeler. And okay, what is this? Okay, I selected the query sequence again, just skip it. And now we have total number of files is 6. 4 structure files, 1 pdb underscore 95.pir file, and 1 the query sequence.ali file. Now I'll just go on the modeler, and here you can see that at above it's written that you can for the to run we need to write mod 9.25. So first I'll go into the bin folder where I've stored all the files, and in bin that one modeler 1 folder, click enter. Now I am inside this folder, and now I will run the command for the model for structural modeling. So first what you need to do is you need to have, as I just showed you in the beginning, that we need to install Python, because there are defined scripts which are already available while you will download the modeler. These scripts actually contains the aligning, building, evaluation, everything. So there are 3-4 main scripts. So first the script which we need to use is the build underscore profile light.py. So I will also paste that script inside the folder where my other files are placed, and here using any text editor you have, just open this Python script, and we will change a bit of that thing. Nothing will change in the syntax or in the commands or anything. We will just replace the we'll just replace our alignment file, ALI file, which we have made. That is for the first thing what we do is we will change the query sequence.ali here from the example of file we downloaded for the modeler. So query sequence.ali, right? Now it's changed, so let's every other thing is here. Now let's go on the let me see if the things are going in the right direction. Example file which we downloaded, check the aligned. So these are the files they used. So this is the example, and here build underscore profile, they also used it. And these are the, then it will also, these are the output files. So these are actually are the predicted structures after the performing the modeling. So let's see what we can do with our analysis, right? So now we are in the modeler 1 folder, so we will just write mod 9.25 space, then we'll give the command here. What command we need to do is query sequence. No, sorry, we have to give the command of the Python script. So what is the name of the file? We just go and check it. For example, in the program files, users, here I'll just go and check the name of the file in the bin folder, modeler 1. So the name is the build underscore profile.py. So I will write this here, right? Build underscore profile, right, dot py. So this is the first command. There are multiple other scripts as well, what we will use for the analysis, and by the end of the running all these 3-4 commands of the 4 scripts, we will be able to have the structures, and we will also evaluate those structures. So the protein amino acid sequence was also very large, that was around 26,000 amino acids, so it will take a bit of time. And in the next part, so here we are done. So here we can say that we have successfully installed the modeler. We have successfully made all the formats we have to use for performing the modeler, and also we have modified the Python script by changing the our alignment file, and now we have run the first build underscore profile.py command. So in the next part, we will come up with the other commands as well. So let's wait for the next part. In the meanwhile, please go and reproduce these all things on your own computer. So here build underscore log file is now building in Rest. Thank you. And if you have any questions, you can ask in the question bar.